Good morning, ma'am. Good morning, everyone. Myself, Manshankar, pursuing MSc Microbiology, Central University of Tamil Nadu. Today, I am going to talk about Sponge Microbial Association. Here, Sponge Microbial Association, nothing but symbiotic relationship between the sponge and the microorganisms. Uh, Now, look into the topic. So, you can see here a table of content I am going to discuss in this topic. You can see here. Coming to the introduction part. Here the sponges are the mainly hotspots for the microorganisms, and sponges are multicellular organisms are lacking the true tissue organs. The sponges contain the spores around the tissues, so it filter out all the water and trap the nutrients. Sponges are the diverse microorganisms group here, bacteria, archaea, and the fungi. These are the association for the mutualism, meaning the both organisms are benefiting. And the sponges mainly found in the marine as well as the freshwater for the nutrient cycling. This is about the small introduction about the sponge and microbial association. Now coming to the types of sponge microbial association, there are different types of association among the sponge and microorganism. So we will discuss some of them. Here the endosymbiotic bacteria. There are different type of symbiotics are there, endosymbiotic, ectosymbiotics, like that. So we are discussing endosymbiotic. Endosymbiotic mean nothing but the microorganisms lives inside the tissue of the sponges. So it plays a major role in the nutrient cycle detoxification. Coming to the epibonites are the microbes that resides in external surface of the sponges. So it is also involved in the formation and protection against pathogens. Now coming to the pathogen association, some microorganisms may access as pathogens in the specific environments, um, causing disease in sponges. And the commercialism here, the microbes that benefit from the living in the sponges without significant effectance. This about the sponge microbial association, types of sponge microbial association. And see the symbiotic relationship. The symbiotic relationship between the sponge and the microbes. Here the sponges are the host to the microorganisms. Wherever the sponges are there, microbes that we command survive in the tissue of the sponges, and these associations often for the mutualism. Here the both sponges microbes are the benefiting, and microbes help to the sponges in the various ways. Example here, the nutrient cycle in the production, while the sponges provide the stable habitat and the nutrient for the microbes. In this way, symbiotic relationship between the sponge and the microorganisms. We can see in these slides microbial benefit to the sponges. How the microorganisms are benefiting to the sponges. Here the nutrient acquisition. Microbes break down the organic matter and the cycle nutrients like nitrogen and carbon, making them to accessible to the sponges. Here the mainly microorganisms are breaking down the organic matter. So like nutrients like nitrogen and carbon access to they easily sponges are accessing. And coming to the detoxification, microbes detoxify harmful waste products, ammonia preventing sponges tissue damage. See here the ammonia mainly it damages the sponge tissue. Here the microbes detoxify that harmful waste products uh, and preventing the sponge tissue. Here the chemical defenses. Symbiotic microbes are produced secondary metabolites like antimicrobial compounds. These sponges from the pathogens like predators. Here the mainly symbiotic microbes are produced secondary metabolites. It releases some antimicrobial compounds. These are protecting the sponges. In this way, the microbes are benefiting the sponges. In the last slide, we seen how the microbes are benefiting to the sponges. Here we can see how the sponges are benefiting for the microorganisms. Here the mainly sponges are providing the stable environment because the microbes are microbes are surviving inside the tissues of the sponges. Here it provides a stable and environmental surface and for microorganisms to grow and reproduce, are protecting from the predators and the harsh environmental condition. Here in this way, sponges are benefiting for the microorganism and as well as access of nutrients. Sponges filter the large volumes of the water, uh, capturing for the food particles and the organic matter that are associated microorganisms. Can utilize the associated microorganisms. In this way, here the sponges already said you that sponges contain the spores. From the spores, it filter out all the nutrient. That nutrients are microbes are taking that nutrients as a food and surviving. In this way, these sponges are benefiting for the microorganism. Also, symbiotic interactions. Some microorganisms may be benefit from the metabolic products of the byproducts of the sponges, creating the mutual benefit of exchanging of the resources. In this way, uh, sponges are benefiting the microorganisms. Here we can see in this slide high microbial abundance, HMA. High microbial abundance is nothing but sponges with a high density of microbial cells. The sponges contains more microbial cells on the tissue surface, composed of 40% of sponge biomass, compressing up to 40% of biomass. HMA, high microbial abundance, having more complex with the dense tissue. It having the more complex and the tissue are will be dense because of the lot of microbial content in the tissues. So the structure there be a slower filtration. So in the high microbial abundance, it filters slowly of the water as well as the nutrients. So metabolites production. Microbial symbiotes in the HMA sponges often responsible for the producing the bioactive compounds, including the bioactive compounds are nothing but including the antibiotics, toxins, and the antifungal agents. It role of ecosystem, high HMA, role of HMA in the marine ecosystem. HMA sponges are important for the marine ecosystem and the biological cycling and the nutrient recycling. This is about the high microbial abundance. Coming to the low microbial abundance. In the last slide, we discussed about a high microbial abundance. Here in the low microbial abundance, the microbial load is less compared to the high microbial abundance. So here the filtration is faster compared to the high microbial abundance because of the tissues are the less congested with the microbial cells. Low microbial abundance sponges are the fewer microbes, so it produces a few bioactive compounds, and it found the more nutrient rich environment because it for the faster filtration, uh, it directly capture more nutrients. It is the advantages for the low microbial abundance compared to the high microbial abundance. Coming to in this slide, we can see here biogeochemical roles. It's the mainly microbes are doing nitrification. Microbes that convert the ammonium into nitrate, and that nitrate which are the less toxic and can be used for the other marine organisms. Here, microbes are not only benefiting for the sponges as well as is benefiting for the marine organisms also. Coming to the denitrification, microbes help in reducing the nitrate back to the nitrogen gas, maintaining the nitrogen balance in the marine ecosystem. So, by the doing the denitrification process, it balances nitrogen in the marine ecosystem. Coming to the methane oxidation, microbes are involved in the methane cycling, helping to reduce the greenhouse gases in the ocean environment. In this way, biogeochemical roles, the microbes are helping not only the sponges as well as the marine ecosystem. So in this slide, we can see key microbial functions in the sponges. We can see how the microbes, what are the functions of the microbes in the sponges in this slide. And here we can see nutrient acquisition. Nutrient acquisition is nothing but microbial assist in the breakdown of complex organic molecules into the simpler forms, so that sponges can be easily absorb that organic matter, and after that it will do the production of the bioactive compounds like antibiotics and the antifungal agents. When it goes to the stress condition, it produces these bioactive compounds. After that, conversion of the waste products into the useful nutrients. This mainly done by the microbes, as well as microbes produce the toxins or unpalatable substance to detector and predictors. So these are the key microbial functions in the sponges. So in this slide, we can see sponge and bioactive compounds. How the bioactive compounds are formed in the sponges. See, when the marine sponge is formed as a defense mechanism in the response to the stress, factors like predictors and the competition for the space. When it's have this stress condition due to the predictors and the competitions, so it releases the secondary metabolites. From that, the bioactive compounds are being formed. If these bioactive compounds are useful for the pharmaceutical importance, like from that it bioactive compounds, from that it produce like antibacterial, antiviral, antifungal and the anti cancer properties are there in the bioactive compounds. You can see example here, anti-cancer agents from sponges are used in preclinical and the clinical trials for the cancer treatment. Example is helichronium bead derived from the helichronidia sponges. It basically acts as anti-cancer drug in the elbrulin. And the antimicrobial agents, sponges is a source of the novel antibiotics, especially due to the rising antibiotics. Example here, azilipron, isolated from the sponges, show activity against the multi-drug resistance bacteria. So this support the sponge and the bioactive compounds. So in this slide, we can see factors that influencing the sponge and microbial association. We can see there are some factors that environmental condition, host-immune system, and the water depth and the biofilm formation. We can see the environmental condition factors that has temperature and pH, salinity can be affect the microbial community. These are different temperatures and the different pH conditions that affect the growth of the sponges, as well as the host immune system. Sponges as a primitive. Primitive means it's not developed. It's a normal immune system that can be regulate the microbial load and the diversity and the tolerance certain microbes. As well as we can see are the water depth. Microbial association can change the depth. Differences in the light availability, pressure, and the temperature. Here the due to the depth of the water as well as the light that penetrate into that water, that also factors that influencing the sponge and microbial association. And the finally biofilm formation. Microbes within the sponges may form the biofilms which contribute the stability and the structural microbial communities. These are the main factors such as influencing the sponge and microbial association. So in this slide, we can see research on the sponge microbial communities. Here, for this identifying that they use some techniques. We can see here the metagenomics. Metagenomics researchers use the metagenomic sequence to identify the microbial species and their functional genes within the sponge's tissue. They will using the metagenomics, they will identify the antimicrobial compounds as well as genes within the sponge tissue. Here they will take the sponge sample and they done the metagenomics. They isolate the DNA from the sponge sample. They construct the metagenomic library, and they use a PCR based screening sequencing. From there, they identify the genes. From the gene, they identify the antimicrobial compounds. This is an example for the metagenomics. As well as they will do the 16S RNA sequencing. In this method, they helps to identify the classify the bacterial communities based on the 16S RNA. The using of 16S RNA, they classify the bacterial communities, and proteomics and the cryptomyces. These are the approaches helps to study expression of the proteins and the gene within the sponge microbial communities and revealing the metabolic functions and the interaction and the symbiotic evolution. Researchers show that sponge microbial associations have like evolved over millions of years with their mutual benefits. By these methods, they used to do the microbial communities, sponge microbial communities. So here comes to the conclusion point. Sponge and microbial association essential for the survival of both sponges and the microorganisms. Here the sponges contain the spores in the tissue. It filter out all the nutrients throughout the water, and the nutrients are essential for the microbial growth. And here the sponges produce the secondary metabolites from the bio as well as the biofilms. From that, antimicrobial agents like antifungal and antiviral agents are produced. The compounds lead to discovery of new compounds for the medicines. This both sponge and microbial association may shed light to the marine ecosystem and the healthy biodiversity. This is about the conclusion of sponge and microbial association. So these are my references. Thank you, ma'am, for giving this opportunity, and thanks to everyone. Thank you.